Dear friends, once again hearty and warm welcome. Friends, in this lecture we are going to learn new topic, that is experimental evidence for semi-conservative replication of DNA. Dear friends, we have already studied about the meaning of replication and also the mechanism of replication. We know that replication is a process in which parent DNA produces two daughter DNA molecules which are exact copies of itself. Replication is a process in which parent DNA produces two daughter DNA molecules which are exact copies of itself. Now, the concept of replication, idea of replication was first of all suggested by Watson and Crick. Watson and Crick suggested that the DNA undergoes replication, duplication, during which the double-stranded DNA undergoes unwinding, uncoiling. It means the two strands of DNA unwind, uncoil. It occurs the unzipping so that the two strands get separated from each other. Now that each strand acts as a template strand or mold strand on which the new complementary strand is synthesized along it. It means that on each old strand, the new complementary strand is synthesized. And at the end of process of replication, two daughter DNA molecules are synthesized. Each daughter DNA molecule receives one old strand and one new strand. It means each of the daughter DNA molecule receives one parental strand, that is half of the material, half of the DNA, or we can say one strand of parental DNA is conserved, and therefore this method of replication is also known as semi-conservative method of replication. Now, this semi-conservative method of replication in eukaryotic DNA was proved first of all by scientist Taylor in year 1957. युकॅरियोट्स मध्ये डीएनएचं रेप्लिकेशन हे कशा पद्धतीने होतं हे टेलर या शास्त्रज्ञाने एक्सपेरिमेंटली प्रूव्ह केलं. व्हाईल द टू डिफरंट सायंटिस्ट, नेमली Meselson and Stahl, Matthew Meselson and Franklin Stahl, they performed experiment, and through their experiment they gave a very strong support to the semi-conservative mode of replication in prokaryotes by performing experiment on Bacterium Escherichia coli. Bacteria Escherichia coli. याचा अर्थ मेसेल्सन आणि स्टाल या दोन शास्त्रज्ञांनी ई कोलाय, इस्करिशिया कोलाय या बॅक्टेरियमवर प्रयोग करून त्या ठिकाणी डीएनएचं रेप्लिकेशन प्रोकॅरियोट्स मध्ये सेमी कॉन्झर्वेटिव्ह याच पद्धतीने होतं हे प्रूव्ह केलं, हे सिद्ध करून दिलेलं आहे. And now therefore we people have to study about that Meselson and Stahl experiment in support of semi-conservative mode of replication. Meselson and Stahl's experiment. Meselson and Stahl, they performed this experiment by using one special technique, namely equilibrium density gradient centrifugation technique. Listen carefully. Meselson and Stahl, they performed that experiment to prove the semi-conservative mode of replication by using a special technique known as equilibrium density gradient Centrifugation technique. Centrifugation technique. First of all, let me teach you little bit more detail about this technique, and then we shall learn about the actual experiment performed by Meselson and Stahl. This equilibrium density gradient centrifugation technique developed by Meselson and Stahl enable the scientist to separate different substances in the mixture. Try to understand. This technique developed by Meselson and Stahl enables the scientist to separate the different particles, different substances in the mixture. Now, what this technique is, let me explain you. Here, the title of this technique, name of this technique is equilibrium density gradient centrifugation technique. All of you are known what is density. Density म्हणजे काय हे आपल्या सगळ्यांना माहिती आहे. Then and then let us talk. We know that density, it can be defined as the amount of matter in unit area, or we know that the density, it can be represented as mass per volume. Mass per volume. याचा अर्थ युनिट एरिया मध्ये एखाद्या पदार्थाचं प्रमाण किती आहे यावर त्याची डेन्सिटी, घनता ही अवलंबून असते. इफ इन अ युनिट एरिया द अमाउंट ऑफ मॅटर इज मोर, then obviously that material has high density. एखाद्या विशिष्ट युनिट एरिया मध्ये, एकक क्षेत्रफळामध्ये जर एखाद्या पदार्थाचं प्रमाण, अमाउंट जर जास्त असेल तर अर्थात त्या पदार्थाची घनता, डेन्सिटी ही जास्त आहे. and if the amount of any matter in any unit area is comparatively less, then that material has less density. याचा अर्थ युनिट एरिया मध्ये कोणत्याही पदार्थाचं अमाउंट किती आहे, प्रमाण किती आहे याच्यावर त्याची डेन्सिटी ही ठरत असते. नाउ हियर Meselson and Stahl performed this experiment for which they used the centrifuge machine. Let me explain you. Centrifuge or centrifugal machine, it is an instrument which exerts centrifugal force on the material in the test tube or in a container. Listen carefully. The centrifuge, it is an instrument which exerts centrifugal force on the material used, or centrifugal force on the material present in the container, in the vessel, or we can say test tube, which are placed in that centrifuge machine. Actually, this centrifuge machine, its rotor spins that material very rapidly, and because of which the centrifugal force is exerted on the material, and that centrifugal force, it moves the material away from the center and bottom of the test tube or bottom of the vessel. Let me explain you properly. Just consider, here is the centrifugal machine. Diagrammatically I am going to explain you. कल्पना करा की या ठिकाणी कुठलं तरी सेंट्रीफ्युगल मशीन आहे. Now in this centrifugal machine, here is a rotor with central rod, and in this rotor, suppose the provision is made to place the 4 test tubes. Consider, these are supposed to be the test tube placed into this rotor. Okay? The material which is to be centrifuged, the mixture which is to be centrifuged, ज्या मिक्सचरचं या ठिकाणी centrifugation करायचंय, that mixture of substances is filled in these test tubes. हे मटेरियल या test tubes मध्ये भरलं जातं. These test tubes are placed here in this machine, and when this rotor rotates at very high speed, then obviously the material present in the test tube experiences the centrifugal force, and because of which the materials present in that mixture experience the centrifugal force according to their density, and either they settle down at the bottom of test tube or may comes up as a supernatant. Of course, that centrifugal force experienced by the materials in the substance is depend upon density of that material. If the material is with high density, then it experiences greater centrifugal force and it settles down, while the material with less density, it experiences less centrifugal force and it remains at upper level or as a supernatant in that mixture. मला काय सांगायचं आपल्याला कळालं असेल. या ठिकाणी मी सेंट्रीफ्युगल मशीनचं डायग्रामॅटिक रिप्रेझेंटेशन केलंय. या ठिकाणी जो रोटर आहे त्या रोटर मध्ये अशा प्रकारे टेस्ट ट्यूब्स किंवा व्हेसल्स फिक्स करण्यासाठी प्रोव्हिजन केलेली असते. ज्या मिक्सचर मधल्या पदार्थांना आपल्याला सेपरेट करायचे, वेगळं करायचे, ते पदार्थ, ते सबस्टन्सेस या टेस्ट ट्यूब मध्ये फिलअप केले जातात, भरले जातात. आणि ज्या वेळेला हा रोटर या ठिकाणी स्पिन होतो त्यावेळेला अर्थातच या प्रत्येक टेस्ट ट्यूब मध्ये असलेल्या मटेरियलला सेंट्रिफ्युगल फोर्स हा एक्सपिरियन्स होतो आणि त्यामुळे ज्या पदार्थांचं डेन्सिटी जास्त आहे त्यांना सेंट्रिफ्युगल फोर्स हा जास्त एक्सपिरियन्स होतो आणि ते सेटल डाऊन होतात. अँड दोज मटेरियल्स विच हॅज लेस डेन्सिटी दे एक्सपिरियन्स लेस सेंट्रिफ्युगल फोर्स अँड दे रिमेन सुपरनटंट. अर्थात या ठिकाणी इन दिस एक्सपेरिमेंट द स्टँडर्ड सेंट्रिफ्यूज मशीन्स आर नॉट युज्ड बट अल्ट्रा सेंट्रिफ्यूज मशीन्स आर युज्ड. या टेक्निक मध्ये अल्ट्रा सेंट्रिफ्यूज मशीन्स यूज केले जातात. That ultra centrifuge machines spin at very high speed, about 5 times more than the standard centrifuge. सर्वसाधारण जे काही सेंट्रिफ्यूज मशीन्स आहे त्याच्यापेक्षा हे अल्ट्रा सेंट्रिफ्यूज मधलं जे स्पिनिंग आहे ते 5 पटीने जास्त असतं. And because of which the materials present in the mixture not only get separated from each other but also there is a formation of density gradient. लक्षात घ्या, याचा अर्थ हाय स्पीड स्पिनिंग मुळे त्या मिक्सचर मध्ये असणारे वेगवेगळे पदार्थ फक्त एकमेकांपासून वेगळे होत नाही तर त्या ठिकाणी आपल्याला क्लिअर कट डेन्सिटी ग्रेडियंट दिसतो. डेन्सिटी ग्रेडियंट याचा अर्थ पण मी सांगतो. डेन्सिटी ग्रेडियंट मीन्स सिंपली it is the variation in the density of given area. विशिष्ट ठिकाणी असणाऱ्या पदार्थांच्या डेन्सिटी मध्ये असणारी विविधता, डिफरन्स, फरक, व्हेरिएशन याला म्हणतात डेन्सिटी ग्रेडियंट. आपण कल्पना करू शकतो. एक मी छोटंसं जनरल उदाहरण देतो. जस्ट कन्सिडर सपोज वी हॅव अ बीकर, ग्लास बीकर. आपल्याकडे एखादं काचेचं बीकर आहे, भांड आहे. कल्पना करा, दॅट इज फिल्ड बाय मीन्स ऑफ वॉटर. त्याच्यामध्ये आपण पाणी घेतलेलं आहे अँड इन दॅट वॉटर वी हॅव मिक्स्ड अ फाईन पावडर ऑफ ग्लास. काचेची बारीक पावडर, भुकटी आपण त्यात टाकली. याच्या व्यतिरिक्त इफ वी ॲडेड से द पावडर ऑफ चॉक, चॉकची पावडर आहे आपण त्याच्यामध्ये मिक्स केली अँड वी स्टर्ड दिस एंटायर मटेरियल. आफ्टर स्टरिंग, आफ्टर स्पिनिंग ऑफ दॅट मटेरियल, what will happen? We know the glass particles will settle down and the dust particle will come up. It means that it occurs the separation of these two materials in that mixture depending upon their density and only because of the centrifugation. This technique basically used by the Meselson and Stahl for the experiment to prove semi-conservative replication. Now we have to study the actual experiment. आता प्रत्यक्ष आपण त्यांनी एक्सपेरिमेंट कसा केला हे समजून घेणार आहोत. Now we know that the DNA is nothing but the polynucleotide molecule. It means DNA is made up of two long chains of deoxyribose nucleotides. Each deoxyribonucleotide consists of nitrogen base, We know that in DNA, 4 different types of nitrogen bases are present, such as adenine, guanine, thymine, cytosine. Nitrogen bases are nothing but the nitrogen containing organic salts. It means that nitrogen is the main component of DNA. याचा अर्थ नायट्रोजन हा DNA चा मुख्य घटक आहे. Normally, 2 different isotopes of nitrogen are found in the nature. निसर्गतः नायट्रोजनचे 2 isotopes आढळतात. Let me explain you. Such as the N14 and N15. N14, it is the light isotope, and N15 is heavy isotope. Mind well, though the N15 is heavy isotope, it is non-radioactive. Okay? Now what does it mean? And what these figures indicate, 14 and 15? These figures indicate the mass number. You know that mass number means the sum of protons and neutrons. Now here in N14, the nucleus has 7 protons and 7 neutrons, and that's why the mass number of this nitrogen, light isotopic nitrogen, is 14. But here in N15 there is one more neutron, that is 7 protons and 8 neutrons, and that's why its mass number is 15. Obviously N14 is light isotope, means it has less density, and N15 is heavy isotope, therefore it has more density, greater density than the N14. Now in DNA and in all biomolecules, abundantly N14, this type of light isotope of nitrogen is found. सर्वसाधारणपणे DNA मध्ये आणि इतरही बायोमोलेक्युल्स मध्ये आढळणारा जो नायट्रोजन असतो तो या प्रकारचा असतो. याचा अर्थ लाइट आयसोटोपिक नायट्रोजन असतो, N14 असतो. बट N15 इट इज ऑल्सो फंक्शनल आयसोटोप, म्हणजे N15 हा सुद्धा फंक्शनल आयसोटोप आहे. Now let us come back to the actual experiment performed by Meselson and Stahl. Meselson and Stahl performed experiment by using bacterium Escherichia coli, E. coli. You know Escherichia coli, it is the bacterium inhibiting human colon. Now initially in first step, the Meselson and Stahl, they cultured E. coli bacteria in the culture medium containing N14. Listen carefully. सुरुवातीला त्यांनी E. coli या बॅक्टेरियांना N14 असलेल्या कल्चर मीडियम मध्ये ग्रो केले. फॉर सेव्हरल जनरेशन, नॉट ओनली फॉर वन जनरेशन, सेव्हरल जनरेशन. सो I said that Meselson and Stahl initially cultured the E. coli bacteria in the culture medium containing N14 for several generations, so that all bacteria produced after several generations would have only N14 as the component of DNA. जेणेकरून अनेक जनरेशन नंतर असे बॅक्टेरिया तयार होतील की ज्यांच्यामध्ये डीएनएतला नायट्रोजन हा फक्त याच प्रकारचा असेल. याचा अर्थ N14. Now after several generations they collected that bacteria from this culture medium and ultracentrifugation is carried out. त्यांचं ultracentrifugation हे घडून आणलं. Look, due to ultracentrifugation in the 6 molar cesium chloride अर्थात या ठिकाणी कलेक्ट केलेले जे बॅक्टेरिया आहेत त्यांना सेंट्रीफ्यूज करण्यासाठी त्या ठिकाणी मीडियम म्हणून 6 मोलर सीसियम क्लोराइड याचा वापर केला. And after centrifugation they found a density gradient band somewhat at the upper level of the test tube. Somewhat at the upper level of test tube. Try to understand. I said that Meselson and Stahl, they cultured E. coli bacteria in the medium containing N14 for several generations. Then they collected that bacteria all with N14 in their DNA, and that bacteria were centrifuged in centrifuge machine in a medium of cesium chloride. And after centrifugation they found the density gradient band of DNA at upper level in that mixture. Consider this is supposed to be that cesium chloride. कल्पना करा की या ठिकाणी हे cesium chloride मीडियम आहे आणि सेंट्रीफ्युजेशन केल्यानंतर डीएनए मॉलिक्युल्सचा जो डेन्सिटी ग्रेडियंट बँड तयार झाला, थर तयार झाला, लेयर तयार झाला, हा सर्वसाधारण या ठिकाणी टेस्ट ट्यूबच्या अप्पर लेवलला तयार झाला. Now they transferred these bacteria containing N14. Now they transferred these bacteria containing N14 into another culture medium containing N15. Here is the culture medium with N15. I said that in second step they transferred the E. coli bacteria with N14 into another culture medium containing N15, that is heavy isotope of nitrogen, and allowed for multiplication for several generations. याचा अर्थ त्या N14 असलेल्या बॅक्टेरियांना N15 असलेल्या कल्चर मीडियम मध्ये ट्रान्सफर करून त्यांचं अनेक पिढ्या मल्टिप्लिकेशन होऊ दिलं, जेणेकरून त्या ठिकाणी आफ्टर सेव्हरल जनरेशन्स whatever bacteria will produce, that all with N15 and not N14, because now during multiplication that bacteria utilized the nitrogen from this culture medium which was heavy isotope, and that's why the DNA of all those bacteria were with heavy isotope, that is N15. N14 get replaced by N15. Now these bacteria collected and again centrifuged. आता या ठिकाणी हे बॅक्टेरिया त्यांनी कलेक्ट केले आणि त्यांचं परत सेंट्रीफ्युजेशन घडवून आणलं. In the same medium, that is nothing but the, we can say, cesium chloride. At this time, they found the density gradient band near the bottom of test tube. Near the bottom of test tube. Please note, when they centrifuged the bacteria with N14, the density gradient band appeared at upper level in the test tube, while when they centrifuged the bacterium with N15, the density gradient band appeared at the bottom. Right? Why this is so? Because it indicated the N14 has low density and N15 has high density. Now, in next step, they transferred the E. coli bacteria containing N15. आता पुढच्या स्टेप मध्ये त्यांनी N15 N15 असलेले जे बॅक्टेरिया आहे ते परत N14 असलेल्या कल्चर मीडियम मध्ये ट्रान्सफर केले. N14 असलेल्या कल्चर मीडियम मध्ये ट्रान्सफर केले. कन्सिडर करा, इन दिस वेसल हियर इज अ N14 in the culture medium, and they collected the bacteria produced in first generation just within 20 minutes, because the life cycle of bacteria is completed within 20 minutes. बॅक्टेरियाचं लाईफ सायकल 20 मिनिटात संपतं, अर्थात त्याची नवीन पिढी ही 20 मिनिटात तयार होते. Now when they transferred the bacteria with N15 again into the culture medium with N14, after 20 minutes the bacteria of first generation were collected. Bacteria of first generation were collected and they were centrifuged again in the same cesium chloride solution, and at this time they got the density gradient band. Listen, at this time they got density gradient band not at the bottom like N15 or not at the top like N14, but at this time the density gradient band of the DNA appeared exactly at the middle. Exactly at the middle. Let me show. Exactly at the middle. Now what does it mean? It indicates that the DNA of bacteria produced in first generation neither contain only N15 nor N14, but obviously the 50% of N14, 50% of N14, and that's why the density gradient band appeared at the equal distance from the level of N14 and N15, that is exactly at the middle. It suggests that the bacteria produced in first generation contain hybrid DNA. That is, the 50% of DNA with N14 and 50% of DNA with N14. Okay? Explain करतो. कल्पना करा, या ठिकाणी हा जो N14 मध्ये कल्चर केलेले बॅक्टेरिया होते, त्यांच्यामध्ये आपल्याला माहिती आहे की दोन्ही जे स्ट्रँड्स आहेत DNA चे, त्याच्यामध्ये N14 हे प्रेझेंट आहे. या ठिकाणी जे बॅक्टेरिया सेंट्रीफ्यूज केले त्यांच्यामध्ये DNA च्या दोन्ही स्ट्रँड मध्ये N15 हा हेवी आयसोटोप आहे. आणि आता या ठिकाणी या फर्स्ट जनरेशन मध्ये तयार झालेले जे बॅक्टेरिया आहे त्यांच्यामध्ये मात्र डेफिनेटली DNA मध्ये 50% N14 आणि 50% N15, याचा अर्थ one strand of DNA is with N14 and one strand of DNA is with N15. That is semi-conservative replication, isn't it? Now again, to confirm this semi-conservative replication, they further cultured that bacteria of first generation into the culture medium containing N14 again. Look, या ठिकाणी फर्स्ट जनरेशन मध्ये तयार झालेले जे बॅक्टेरिया आहे की ज्यांच्यामध्ये अशा प्रकारचा आता हायब्रिड DNA, isn't it? This is hybrid DNA. Because this hybrid DNA has 50% N14, 50% N15. Now such bacteria with hybrid DNA produced in first generation, when cultured again into the culture medium with N14, Then at this time the second generation of bacteria showed some another result. या ठिकाणी सेकंड जनरेशन मध्ये तयार झालेले जे बॅक्टेरिया आहेत त्यांच्याबाबत वेगळे रिझल्ट्स मिळाले. When that bacteria of second generation collected, centrifugation carried out, at that time two different density gradient bands appeared. दोन प्रकारचे डेन्सिटी ग्रेडियंट बँड त्या ठिकाणी निर्माण झाले. कोणते? लेट अस सी. One band exactly at the middle, one band exactly at the middle. You know, this band at the middle represents hybrid DNA. म्हणजे हा जो बँड तयार झाला तो हायब्रिड DNA चा आहे. And second band it appeared at the upper level, means at the level of N14, at the level of N14. दूसरा बँड हा अप्पर लेवलला तयार झाला N14. What does it indicate? The bacteria of second generation contain 50% of the hybrid DNA and 50% of the DNA with only normal N14. याचा अर्थ आता या सेकंड जनरेशन मध्ये तयार झालेल्या बॅक्टेरियांमध्ये 50% DNA हे हायब्रिड आहे या प्रकारचं आणि 50% DNA हे नॉर्मल नायट्रोजन दॅट इज N14 या प्रकारचं आहे. याचा अर्थ काय होतो? लेट अस सी. या ठिकाणी आपल्याला माहिती आहे की फर्स्ट जनरेशन मध्ये जे बॅक्टेरिया तयार झाले त्यांच्यामध्ये हायब्रिड DNA आणि आता असा हायब्रिड DNA असलेल्या बॅक्टेरियांना ज्या वेळेला पुन्हा N14 असलेल्या कल्चर मीडियम मध्ये ग्रो केलं जातं त्यावेळेला काय होईल बघा. You know that during replication the two strands of parent DNA separate from each other. So these are supposed to be the separated strands of hybrid DNA, and on this hybrid DNA the new strands are synthesized. But for the synthesis of new strand on old strand, parental strand, the material used from this culture medium containing N14. So obviously here is the new strand form is with N14, what I have shown with the help of yellow chalk, and here also the new strand synthesized with N14, again going to show with the help of yellow chalk. याचा अर्थ या ठिकाणी काय झालं की दोन प्रकारचे DNA तयार झाले. 50% of the hybrid DNA and 50% with only N14. हे काय शक्य झालं? This became possible only because of the semi-conservative method of replication. जर थर्ड जनरेशनचा आपण अभ्यास केला तर continuously the percent of hybrid DNA goes on decreasing and percent of normal DNA or DNA with normal nitrogen N14 goes on increasing. या ठिकाणी सेकंड जनरेशन मध्ये हायब्रिड DNA 50% आहे आणि नॉर्मल DNA 50% आहे. थर्ड जनरेशन मध्ये जर गेलं तर हा रेशो हायब्रिड DNA चा होणार आहे 25% आणि नॉर्मल DNA चा होणार आहे 75%. So this entire experiment clearly proved that in prokaryotes, in Escherichia coli bacteria, the replication is of semi-conservative method. It means during replication, each daughter DNA molecule receives only one parental strand and one new strand. That's all about this experiment, and here ends this topic. Thank you.